and how it regulates the metabolism and also ROS dynamics of plants. So first, I'm going to start off by giving you a short introduction on GABA and what it is. So GABA, or GABA-aminobutyric acid, is a non-proteinogenic amino acid. It is found in all the kingdoms of life, including microorganisms, plants, and animals. And GABA can be synthesized via 3 pathways. The first and the major pathway is via the cytosolic glutamate decarboxylase. And this pathway is depicted here at the bottom. So alpha-ketoglutarate from the tricarboxylic acid is converted to glutamate. Glutamate is then exported from the mitochondria into the cytosol, where it is converted to GABA via the glutamate decarboxylase, or GAD enzyme. Then the GABA is then retransported back into the mitochondria, where it's converted to succinic semialdehyde and succinate, and it can then feed back into the tricarboxylic acid cycle. So in essence, this process at the bottom is called the GABA shunt, and it bypasses 2 steps in the TCA cycle. And these 2 steps, or the 2 enzymes that it bypasses, are very sensitive to oxidative stress. So during oxidative stress, this is a key component to keep the TCA cycle active. Now, the second mode of synthesis for GABA is via the breakdown of polyamines, or the oxidation of putrescine and spermidine. And the last way is still a predicted way. It's not been proven yet. It's a non-enzymatic method from the oxidation of proline under oxidative stress. Now, in plants, in the recent years, it has been suggested that GABA can act both as a metabolite, but also as a signaling entity. So if you move back to animals and what is known in animals, GABA acts as an inhibitor of neurotransmitter signals, and the true GABA receptor has been identified in animals, and GABA is involved in the maintenance of energy homeostasis. So high levels of GABA actually reflect high or low energy availability in animals. But when we move to plants, we believe the opposite is actually true, that high GABA reflects high energy availability. But in plants, GABA has mostly been considered for its metabolic effect and not so much for its signaling effect, until recently, when a GABA binding motif was identified. And this GABA binding motif is situated on an aluminum activated malate transporter. And this transporter causes anion flux across the membrane, so causing membrane depolarization. Typical signaling entities when you have membrane depolarization. And also it's been shown that alterations in GABA levels can affect plant growth. So specifically, it affects the nitrate uptake, it affects photosynthesis, and eventually stimulates growth. And interestingly, it has been shown that isomers of GABA, so non-metabolizable isomers, can inhibit growth, and also that mammalian GABA receptor antagonists are able to inhibit this GABA-mediated growth promotion, suggesting that there might be some correlation between the animal receptor and the putative receptor not yet identified in plants. Now, it has been shown several times that GABA accumulates in response to several biotic and abiotic stresses. And this accumulation is usually associated through calcium calmodulin activation of the glutamate decarboxylase, the cytosolic enzyme that I explained previously. And during these stress conditions, GABA can accumulate to levels much higher than any other free amino acids found in plants, so it's quite significant. And also this accumulation is associated with changes in expression of several genes involved in growth and metabolism of plants. And interestingly, exogenous GABA has been shown to also alleviate all of these biotic and abiotic stresses. And it's mostly mostly function by replenishing the TCA cycle through the GABA shunt, so keeping active metabolism, keeping the TCA cycle running during periods of oxidative stress. But exogenous GABA can also activate the antioxidant responses, such as antioxidant enzymes. And finally, it also induces the accumulation of polyamines. Now, it has been shown in tomato plants that are deficient in abscisic acid. These plants have an overactivated GABA shunt, so they accumulate high levels of GABA. And these plants were extremely resistant against Botrytis cinerea infection. So Botrytis is a necrotrophic fungus. And they also showed that when you treat plants with high exogenous concentrations of GABA, it induces immunity against Botrytis, similar to what is found when you have a high endogenous GABA accumulation. But this is mostly focusing on the metabolic effects of GABA and not really focusing towards the signaling events. So we were more interested in the signaling effects of GABA. So we looked at lower concentrations. And we treated plants with lower concentrations of GABA, and we found that even at 100 micromolar or 1 millimolar, it was comparable to a well-known priming agent GABA. So GABA is beta-aminobutyric acid. It's a structural isomer of GABA, but it's been well characterized to function as a priming agent in plants. And GABA at both low concentrations was comparable to this well-known priming agent. If we look here at the disease severity classes, we can see that for both 100 micromolar and 1 millimolar GABA treatments, the majority of the lesions fell into the lower two classes, which were lesions representative of smaller than 0.3 square centimeters, whereas in the control plants, the majority of the lesions were in the upper two classes, representing lesions larger than 0.3 square centimeters. This is interesting. It suggests that maybe GABA metabolism and signaling can be separated depending on the concentration of GABA present in the specific location of the cell. And to further study this, we looked at the effect of exogenous GABA on the activity of NADPH-oxidases. Now, the NADPH-oxidases are a typical first response when plants perceive pathogens or perceive elicitors of plant pathogens, and they produce a lot of hydrogen peroxide or reactive oxygen species in the apoplast to try and fend off the pathogen. So we treated plants, we pre-treated plants with different concentrations of GABA, and then the next day we exposed them to two different elicitors, flagellin 22, which is a bacterial peptide, and the second one is oligogalacturonides, which is pectin-derived fragments from the cell wall of the plant that gets released upon infection with botrytis. So the first one, when we exposed the leaf discs to flagellin 22, we found that, so this line over here is the control treatment, and the lowest concentration of GABA, it had no effect on the ROS burst peak, but the higher concentrations from 100 micromolar upwards induced a much lower ROS burst peak compared to the controls. So if we look here at the cumulative ROS burst, which is just the total ROS produced over the 60 minutes, we found that at 100 micromolars and 1 millimolar treatment, it significantly reduced the total ROS burst in response to flagellin 22. But once we go up to a higher concentration, like 10 millimolar, this effect was abolished. And this further emphasizes the way that GABA signaling and metabolism can be separated on a concentration-dependent manner. In the past, there was also shown that the expression of NADPH-oxidases was differentially regulated depending on the concentration of GABA present in the roots, and this was during salt stress. If we go back to oligogalacturonides, so the cell wall fragments released upon damage by fungal pathogens, we saw a similar effect, but in essence, we found the lowest concentration of GABA increased the ROS burst peak compared to the control, but at lower concentrations, we also found a reduction in the ROS burst peak. As is clear from the cumulative ROS burst, we found the highest effect for 1 millimolar GABA, reducing it up to almost threefold compared to the control. So this would suggest that GABA would favor a reduced environment rather than an oxidized environment, as it is also known that GABA promotes metabolism and cell survival strategies rather than cell death. So it tries to keep the reactive oxygen species to a low level when exposed to specific stresses. Now that we know that the NADPH oxidase is involved, we wanted to see if GABA has a direct effect on hydrogen peroxide accumulation and also on some reactive oxygen species scavenging enzymes. So we looked at the levels of hydrogen peroxide after treatment with GABA. So we used GABA at 100 micromolars again and 1 millimolar, and we found that this did not have a direct effect on the accumulation of hydrogen peroxide after treatments, but after infection, we did see a significant reduction in hydrogen peroxide content in the treated plants compared to the control. In general, the hydrogen peroxide increases after botrytis infection, as it is a necrotroph that promotes cell death through the accumulation of reactive oxygen species. We also found that both concentrations of GABA was able to induce catalase activity and also ascorbate peroxidase activity, but not guaiacol peroxidase activity. But interestingly, when we infected the plants, all three, the enzymes catalase, ascorbate peroxidase, as well as guaiacol peroxidase, were activated significantly in the pre-treated plants compared to the control. This would suggest that GABA actually primes the defense responses of plants so that they can respond faster and more robustly when they are exposed to a subsequent stress to cope with this accumulation of hydrogen peroxide content. Now, the next step was to see what effect the exogenous GABA has on the metabolism of plants. So we first looked at soluble sugar accumulation after treatment with GABA. So we looked at glucose, fructose, and sucrose levels. So I don't show fructose in this graph because I want to focus on glucose and sucrose and the hexoses. So what we found was after treatment with GABA, glucose and fructose were not significantly affected by GABA directly, but after infection, we found a significant increase in the glucose levels. But the most interesting part here is that sucrose levels increased after treatment directly through GABA. It was some temporal increases at 3 hours and 72 hours, suggesting that we have some sucrose signaling events occurring. And these sucrose signaling events is usually associated with the activation of stress-related compounds, production of stress-related compounds, like anthocyanins and secondary metabolites to prepare the plant for subsequent infection. And interestingly, we found that after infection, we found a decrease in the control plants in both glucose, fructose, sucrose, as well as the total sugar levels. So the soluble sugars decreased in the control plants, but in the plants pre-treated with GABA, we saw actually an increase in the total soluble sugars, suggesting that GABA promotes normal metabolism or try to maintain normal metabolism instead of causing this reduction in soluble sugars. It has also been shown in the past that GABA can promote photosynthesis, so possibly this increase in soluble sugars is due to higher photosynthesis and the ability to maintain functional metabolism during oxidative stress conditions. The next thing we wanted to look at is whether exogenous GABA application can actually induce endogenous GABA synthesis, whether it acts in a feed-forward loop. So we measured the endogenous GABA content, and we found that at 100 micromolar, it had no effect on the endogenous GABA content, so it did not increase endogenously. But when we went up to 1 millimolar, we saw a significant increase in the endogenous GABA production, which reached a peak around 24 hours after treatment and then went down again. And even after infection, we found for both concentrations, GABA accumulates significantly higher in the treated plants compared to the control plants, suggesting that even at a low concentration, GABA can prime its own synthesis, but it needs to be exposed to a stress first, whereas at high concentrations, it directly induces the accumulation of GABA. And it has been shown in the past, actually, that exogenous GABA can also promote the endogenous GABA synthesis, and this happens through the activation of the glutamate decarboxylase, and it's proposed to function through calcium, maybe calcium signatures. And we found that glutamate is the precursor for GABA, but those levels were not significantly affected by GABA treatment, suggesting that the cell tried to balance the glutamate levels from surrounding tissue. It imports more glutamate to keep those levels quite consistent throughout the treatment to supply glutamate for GABA synthesis. So again, this points to a potential effect where we see GABA signaling being split up in signaling events and metabolic events depending on the concentration that you are using. But later on, I'll tell you what exactly we think is going on here. But the last thing that we wanted to look at is whether GABA can also affect nitrogen metabolism. So we looked at the activity of nitrate reductase. So this enzyme is extremely sensitive to periods of stress and also nutrient deprivation because they are controlled by calcium calmodulin kinases, but also by sucrose non-fermenting related kinases or SNRK related kinases. So when they are exposed to a stress, these enzymes usually decrease in activity. They are very, very responsive to stresses. And what we found was that by treatment with 1 millimolar, we found an increase in the total nitrate reductase activity. It includes both the active and the inactive nitrate reductase, but not at 100 micromolar. We did not see any effect after treatment. Whereas when we moved to active nitrate reductase, which only includes the unphosphorylated version, we saw complete different effects where high concentration actually increased the activity, but a lower concentration inhibited the activity, suggesting that at low concentrations, maybe GABA acts as a stress signal, but at high concentrations, GABA is actually depicting a nutrient or enough nutrients in the plant. So it's actually serving as a nutrient source for the plant at high concentrations. But at low concentration, it seems to be a stress signal. And it's also been shown in the past that when plants are starved of nitrogen and you treat them with high concentrations of GABA, it promoted the total nitrate reductase activity. But when there's enough nitrogen availability, there was no effect on the enzyme activity. So a few concluding remarks. We found significant differences between low and high concentrations of GABA treatments. And we showed that at low concentrations, GABA was still able to induce immunity against botrytis cinerea. It was also able to still activate ROS detoxifying enzymes and also to inhibit the accumulation of hydrogen peroxide. It was also able to affect soluble sugar content, specifically after infection. It increased the total soluble sugar content, but it inhibited nitrogen metabolism at low concentrations and promoted nitrogen metabolism at higher concentrations. So when we look at the cellular responses, it also seems to be concentration dependent. It has been shown in the past that different concentrations can affect growth and development different depending on the concentration used, and it's not always a positive correlation. It depends on the tissue type, it depends on the stress condition, and also on the stage of development. So we also found that both carbon and nitrogen metabolism was affected, suggesting that GABA is really central in carbon and in linking carbon and nitrogen metabolism, as is also the case for the TCA cycle. I mean, it links carbon and nitrogen metabolism directly, and GABA can feed directly into the TCA cycle. And the most interesting thing is that exogenous GABA can differentially regulate the activity of NADPH-oxidases, putting it right in the center of defense responses against pathogens. We propose two scenarios. So we propose a high GABA or high exogenous GABA concentrations, where GABA can actually be imported into the cell, probably through similar transporters that is found for glutamate. And upon these conditions, GABA is imported into the cell. It can be fed into the TCA cycle, so used as a food source. And in these conditions, GABA is not perceived as a stress signal, but it actually promotes growth as it represents high energy availability. Whereas at low exogenous GABA, which is probably the case when cells accumulate high endogenous GABA, they get ruptured or they get damaged and they release this GABA into the apoplast, which is then perceived by surrounding cells as danger signals. And they are not imported because the concentration is too low, and they just act as a danger signal to surrounding cells, and they thus promote stress responses instead of growth promoting responses. But these are two different time windows or concentration windows that we are considering for GABA signaling. Thank you for your time. I would like to acknowledge the following people and institutions: